Thank you very much. So we're now transitioning into our panel discussion on the state of research in Maine. So if you haven't taken the opportunity to come forward and would still like to do that, I'd invite you to do that. There are no saved seats up here, so don't worry, you're not sitting in anyone's spot. That spot is for you if you choose to come up and take the spot. My name is Mike McKernan. I'm the Director of Government and Community Relations at the Jackson Laboratory. We are a biomedical research institution based in Bar Harbor with campuses in Ellsworth and Augusta. And that's just in Maine. We have some campuses elsewhere, including Sacramento, California, and in Farmington, Connecticut, and some locations overseas. So thank you, Director Smith and Governor Mills, of course, for organizing this event. And thank you for giving us an opportunity to talk about research today. It's a very important part of the ecosystem that's been mentioned many times this morning already. And so we're glad to share with you exactly how research fits into that critical role into the ecosystem to address this issue that we face state and nationwide. So JAX is a nonprofit biomedical research institution. Our mission is to discover precise genomic solutions for disease, the solutions that are within our DNA. We also empower the global biomedical community to advance their questions in biomedicine. And we do that through creating and distributing worldwide research mice. So mice are elegant model organisms for us to understand human biology, for us to understand human health. And that's one of the things that some of our speakers will talk about today. JAX does not do this alone. We work with partners statewide, and one of those partners is Bowdoin College, and we have Dr. Jennifer Honeycutt here from Bowdoin College. They're one of our education partners in the state. So in addition to Dr. Honeycutt, I'm pleased to introduce my colleagues from JAX. We have, on the left, we have Vivek Kumar. Vivek is an associate professor. We have, next to Vivek, we have Jason Boubier, senior research scientist. And finally, Alyssa Chesler. And Alyssa, Vivek, and Jason are all members of the JAX Center for Addiction Biology. So it's a core group of scientists who work together collaboratively on these issues. And Alyssa is the director of that center. So they'll introduce themselves more fully when they take the podium, but that's who you have with that's who's with us today. So what are our goals for this panel? The first thing we want to do is we want to increase your awareness about Maine-based research to understand the genetic and epigenetic, and what I mean by epigenetic, it's a term that means environmental, largely, origins of substance abuse disorder, including opioids. We want to summarize some data. This will not be a data-intensive set of talks. There will be some data shared. That's what we do. We do research and we gather data and make evidence-based reports of those findings, or report out the evidence that we find. We want to foster dialogue, and so for that reason, we have, well, it was up there earlier, but it'll be in the next slide, an opportunity for you to ask questions using Slido. And Slido is a mechanism you can just call it up on your phone, type in your question anonymously, and I'll see it as the moderator, and we'll use that to direct questions to our panel. We also have a helper, Carol, I believe, who's down here with a microphone. So if you would like your voice to be heard in asking your question, you've got to find your way to this region of the floor. The final thing I want to mention, and then I'll turn it over, is I'm pleased to see that a lot of what I read in the state strategic plan for the opioid response is going to be echoed in what we're talking about today. For example, priority 1, take decisive, evidence-based, focused actions. That's what we do as researchers. We use evidence to better understand biology and better inform future actions, policy, for example, in some cases, other experiments for others. So it's all based in evidence. The other thing that this panel will highlight is priority B of the plan, and that is to prevent early use of addictive substances by children, youth, and young adults, and including reversing adverse childhood experiences, and that's something that Dr. Honeycutt will refer to in her presentation. So that's the format, everybody. That's the goal, at least. The format is each panelist will share their thoughts, We'll come back together as a panel. We'll have an opportunity for Q&A, and we hope the discussion is lively and interactive. There is no bad question. I am not a scientist. I'm not a geneticist. Many of you in the audience may also share that distinction of not being geneticists. It doesn't matter. Please ask your question. These folks are fantastic at answering anything you might put before them. So we'll leave it at that. I would like to now turn it over to Dr. Elizabeth Chesler.
Thank you, Mike, and good morning. I want to talk today about the genetics of addiction, and I want to acknowledge the support of National Institute of Health, National Institute of Drug Abuse, and also the Watson Family Charitable Trust. We've seen this today. It's quite tragic to look at, and what we've seen is increasing rates of overdose death, a slight optimistic cause for optimism in a decline in recent years, and a major reversal of that trend during the pandemic. And this is a disorder of hopelessness. This is a disorder of loneliness. And the question is, what hope can laboratory mice provide us, these quirky mice on Mount Desert Island? I'm going to talk about some of that today. One of the things we know is that many people use and try drugs, but only some of them become addicted. For certain drugs, that's a very large number, and for other drugs, that's a very small number. And we've also heard today that almost no one uses just one drug. People go through a life history of trying different things, and maybe the solution is to try lots of things. And maybe one drug is used to cover up the effects of another drug, and on and on. We get these chain reactions. What we know is among twins that use different drugs, if one twin's using the drug, the other twin is likely to as well. And this explains a large amount of addiction vulnerability. And that vulnerability varies by different substances as well. So we see for cocaine and opiates, very high rates of heritability, the probability that genetically identical twins are going to have similar patterns of use. But it's true for almost every substance, and even for gambling. So we can study this genetic variability, and we can actually use the information from that genetic variability, and we can use that information in both mice and in people, and people do these studies in rats as well. But why bother? Why do this? What do we expect to come from it? And one is actually predicting individual risks of addiction. If we can identify the genetic variants that influence someone's likelihood of developing a substance use disorder, maybe we can alter prescribing practices, especially in particular individuals. Sometimes pain therapeutics are indicated, but maybe for some people they just simply aren't. The risks are too great. We might target preventative interventions. And it's interesting, we can tell you about certain genetic variants, and we can tell you over the entire genome what increases the probability of risk for certain addictions. We can't pinpoint who is going to go down this road. But for a lot of people, they know for generations they have a family history. They don't need me or a genetic test or anything sophisticated to say this is running in our family. But for some people, knowing that that's rooted in biology is a very valuable thing. It takes away some of the stigma and the blame, and perhaps even that self-fulfilling prophecy. But we know that genetics also isn't deterministic. We know that the environment, the life experiences, and many other things can influence the trajectory a person takes, even though they have these genetic predispositions. It's not a done deal. So we don't want people to use genetic information to say, that's it, I'm condemned. I'll just go ahead and hasten the process. We really want to say, there is a biological basis here, and we can use that biological understanding perhaps to move the needle for individuals, identify the states of biology that are consistent with recovery, and those states that are less consistent, and perhaps even induce those states behaviorally or biologically. So we can develop diagnostics and therapeutics based on what we learn about the biology of addiction, identify new molecular targets. And for most disorders, there's some medication that can be used to replace the drug, so methadone, Suboxone. For alcohol, there's several compelling things, including naltrexone. Not widely used, not widely understood, but they work for some people. And for others, like cocaine use disorder, there's currently no therapeutics out there. Some of the other things that we can do is advance our understanding of the disease just generally. What we understand about the biology of addiction can help us understand better what social policies are going to be effective and what might not. They may foster greater understanding and again, more compassion towards people that are suffering. And again, also inform what we do with prevention. Understanding how drugs work in the adolescent brain, how that differs in the adult brain, and how different substances coalesce and contribute to the effects of combined substance use. Most biological research that's practiced around the world in mice relies on a single and very famous strain of mouse, but that's like trying to study all of addiction in one person. What we do at Jackson Lab in particular, and through others using mice that are developed at Jackson Lab, is try to understand from the variability, the diversity of a population of mice, what influences addiction vulnerability. Everyone using a drug is going to have a brain response to that drug. But again, only some people are vulnerable to a substance use disorder from the combined effects of the drug. And it is the case that, for example, with alcohol, you can, even if you're not very vulnerable, through successive exposure, develop an alcohol use disorder, and that's likely true for all other drugs as well. So it's, some people are less vulnerable, but it doesn't mean they're protected. Now, if we look across these different strains of mice, and across males and females, we see differences in almost every addiction-related behavior that we look at, including predisposing behaviors like how likely they are to seek out novel experiences, what their stress sensitivity is, what their daily biological rhythms are. We see that almost all of these have a genetic basis to them. And here we see, so it's novel place preference. That's how much do they like going into a room that they've not been into versus one that they've seen before. A small percentage of that variability is explained by genetics. And we see that a strain with a funny name, PWK, has a very high level of preference for that environment. In the middle, we see the acute response to cocaine. And there again, we see sex differences indicated by the two different lines or points in each color. But we see both sex differences and also strain differences. So strains like WSB, shown in purple, have a very high sensitivity to the effect of cocaine. Again, PWK comes up again, a very high sensitivity, more so in males and females, but still higher than other strains with quirky names like AJ and 129 and B6. And if we look at this thing on the very right, which is how much will they press a lever for cocaine, there again we see that strain that liked to be in a novel environment, that strain that had a high acute response to cocaine, that PWK strain will self-administer a lot of cocaine. And when we take the drug away, that strain continues to seek out the drug. And if we allow it to respond again, we see some reinstatement of that response for the drug. So that's a strain we want to look at for vulnerability to addiction. Most of the research done in the genetics and biology of addiction is actually done in the strain shown in gray here, which responds to drugs but isn't as vulnerable. And if we can compare the differences between these strains, we can find the genes that influence what makes a mouse more vulnerable. And because we share so much of our genetics with humans, over 90%, we can actually identify genes that influence these behaviors in people as well. This is how we do it. When we breed two strains of mice, one that's vulnerable, one that's not vulnerable, in those crossings, in meiosis, you might remember this from high school, we actually end up shuffling the genetic deck of cards, and we can see who's got what cards where in the genome, and that helps us pinpoint where and with increasing precision, which genes are influencing the differences in their behavior. And we can do that now. We've bred over the last 20 years very high diversity populations. These strains have like 90% of all the known genetic variants in commonly used laboratory mice, and we've bred them in different configurations. Each of these pretty colored lines represents what state their DNA is across the genome. So in the inbred strains, we've got PWK is all red, but when we start breeding PWK to the other strains, you could see pieces of their red genome spread throughout each strain. So every strain has some of that PWK DNA, but in different places. And so then we can ask across these mixed up populations, where do they have the genes that are making them vulnerable to various traits? We do this as a group. The Center for Addiction Biology and our colleagues and collaborators around the world are all engaged in studying different traits and different biological mechanisms. But more and more we integrate that data so we can look at vast networks of traits and how they're influenced by different biological characteristics and how they're influenced by genetic variability and its interaction with the environment. We can put all the pieces together and ask questions like, how does hyperactivity relate to drug-seeking behavior? How does impulsivity relate to recovery-related traits? And so we do this data integration. We've also been doing data integration across mice and people, and so we can relate the genetic variation in mice to genetic variation in people, and the genes that are found in mice to genes that are present in people as well. And we use those data in a lot of different ways. It's very hard to collect a large number of people in some of these research studies, especially if you want those people to confess to participating in illegal behaviors. And also when we have a population that doesn't necessarily prioritize or have the means to sign up for these research studies, it's very hard to collect these cases. But what we found is we can identify in mouse genetic studies genes that influence variability in these traits and use the information to enrich our studies of human genetics. And so we've actually shown that with a colleague at Emory University, Rowan Palmer, that we can explain the heritability in tobacco use, cocaine use, and other traits, alcohol use disorder, using genes identified in mice and fish and rats and other species. And this is a big mess, but so is substance use disorders. We've been showing that because each of us are studying different responses to different drugs and different aspects of those responses, we can take hundreds of those traits measured in many different populations and bring all of that data together. And so what we've done here is work with our colleagues in the Psychiatric Genetics Consortium, a very large group of human geneticists. And what we've shown is that we can actually relate what's found in human studies of polysubstance use disorder, which we're looking at the end state of a very complex process. We can say, okay, let's go back into the mouse data and identify what genes that were identified in the human studies are associated with what processes. So we found a gene that was identified in human polysubstance use disorder that indicated, got a role in withdrawal responses to many different drugs in different mouse studies. So now we take this end stage of human polysubstance use disorder and we understand how that genetic variant is acting in, through using lots of aggregate mouse data. So in conclusion, it's super complex. Many genes are involved, also interactions with the environment and life history and things that other speakers will be talking about. But mice and humans share so many genes in common that we can use biology from mouse genetics and data science to learn about the biology of addiction across mice and humans. And this can lead to a heightened understanding of addiction and people with addiction, pointing the way to many different positive outcomes. Thank you.
Hello, my name is Dr. Jennifer Honeycutt. I am an assistant professor of psychology and neuroscience at Bowdoin College, so just up the road. And I started my lab in 2020 and was really excited to get back to Maine, which is where I did my undergraduate work, and to start building my life and family here. So Maine is very near and dear to my heart. And I would love to chat with you a little bit today about some of the research that I do in my lab, as well as contextualize some of what we're talking about within the realm of environmental risk factors. So I'll be sharing how our early life environment, specifically how early life adverse events or traumas, can actually influence how our genes are read and how it can influence behavior later on. So just to kind of preface what I'm talking about, I'm going to mention the adverse childhood. I'm very short.
Sorry. There we go.
Can you hear me better now?
Okay.
Beautiful. Thank you. And I will also try and talk forward because I tend to look behind. But I'm going to talk to you about the Adverse Childhood Experience Scale. So this is a scale that we can use that can tell us something about how many experiences a child has had very early on. So this is prior to the age of 18 that could contribute to negative health outcomes later in life. There are three main kind of categories of ACEs, or adverse childhood experiences. The first is abuse, so things like physical, emotional, or sexual abuse. Neglect is our second one, physical and or emotional neglect, and then household dysfunction, which can range from mental illness of a parent or caregiver, incarcerated relative, a mother treated violently or another caregiver treated violently, substance use in the home, as well as divorce. And so we usually use this, at least clinically in human populations, to understand whether or not an individual may have risk based on prior life experiences for later life negative outcomes. And those negative outcomes can be at the behavioral level. They can also be at the physical and mental health level. So behaviorally, we might see a lack of physical activity, an increase in smoking, alcohol use, other substance use, as well as missed work or other issues with social function. And then on the physical and mental side, there's physiological changes that we see, an increased risk in the likelihood of having different sorts of disorders, different sorts of heart issues, etc. And then in addition to that, we see an increase in risk for mental health outcomes. This particular scale shows depression, but in addition to depression, we see an increase in the likelihood of other sorts of mental health issues such as anxiety, schizophrenia, substance use disorder, the list goes on. As you can imagine, if an individual has multiple ACEs that are built up, you're going to start to see an exponential increase in risk for that individual. In the United States, we tend to look at anything above 2 ACEs as something that's going to start to increase risk. And then as these build, we're going to start to see an exponential increase in that risk as well. So in the lab, I actually focus on one aspect of adverse childhood experiences, and that's neglect. So specifically, I look at psychosocial neglect, so how an individual can navigate their environment in the absence of predictable caregiving. And I do this in a rat model, and so I'll talk a little bit about that. And our behavioral outcomes that we're using are looking at maybe drug use or drug-seeking behavior, which I've done a little bit of in my past work. And now I look and focus a lot more on affective disorders, so things like anxiety and depression, which are also comorbid risk factors for substance use. And so I'll focus on these. And I will preface, I am not a geneticist. So I am actually a behavioral neuroscientist. So a lot of what I do is informed by looking first at behavioral outcomes and then backtracking and trying to understand what is it about this particular environment that an individual might be in, informing a change perhaps to the biology of the organism to then lead to those outcomes. So to contextualize this within the state of Maine, so adverse childhood experiences occur in approximately 14% of children, right? And this is at least what we are able to quantify, where an individual child has 2 or more ACEs. Okay? In the state of Maine, this jumps to 17.9% of our children. Right? So this is a substantial increase. And out of all of the states, so if we look out of 50 states, for example, we are ranked 39, and 50 is the worst, right, in terms of ACE outcome. So we are not ranked well as a state. And so this is a key area that we can focus on to try and understand what is it about things that individuals are experiencing early in life that may pave the way for increased risk. That is not to say that just because you experience an adverse childhood or you have components that would contribute to an ACE score doesn't necessarily mean you are completely at risk, right? I want to be very clear about that, but it does increase your risk. So an early life adverse experience or a multitude of adverse experiences can accumulate over childhood. And we see that an individual with a high ACE score, so 2 or more, for example, are far more likely to develop emotional disorders. And we see, in fact, that this is actually more likely for women and girls in terms of emotional outcomes. And we also see an increased risk for substance use. And we know that both of these independently can lead to an increase in substance use overall, as well as substance use disorder. But importantly, these have an additive effect that really increase the risk even more. So if you have substance use and you have an affective disorder like anxiety or depression, your likelihood is going to then increase. So how do we study this in a rat? So you hear a lot about mice today. I use a rat model. Rats have a beautiful behavioral repertoire, so we can really look at the things that they're doing, how they're interacting with each other socially, their environment, the decisions that they're making. And in my lab, we use a maternal separation model to basically mimic what we see in humans, where we have psychosocial deprivation or a decrease in the predictability of care for these individuals. And this is really modeled off of institutionalized rearing, where there's a very low amount of one-on-one care and predictable care. And what I've seen in the past in some of the research that I participated in as a postdoc at Northeastern University is that our rats that had experienced this psychosocial deprivation over the course of what would be a rat's childhood show an increase in preference for illicit drugs. And so we utilized cocaine, and we used a preference score to try and understand, if given a context, would you prefer the context or the room with cocaine, or would you prefer the context or the room without cocaine? And if you pair these and then ask the animal, which do you prefer, we see that an animal that has had a typical rearing, regular upkeep, they seem to show about a 50-50 preference, right? They'll go to either of those contexts. An individual rat that has experienced early life adversity via this type of separation is going to actually show an increase in their preference for that cocaine paired room. So this is just for one drug. You can imagine that this is also true for many other drugs, and this is replicated in other species. We see this in mice, we see this in rats, and we see this in humans. And so what is it then that we can really understand about what's driving some of this? You'll hear a lot about genetics today, and there's something about biology that can be very comforting, but also can be very scary because you're born with those genes. So if things are impacting our genes, how can we really think about that? We can also think about this in the form of epigenetics. So epigenetics are a means by which our environment, our experiences, our histories, our relationships actually influence how our genes are read, right? So our genes are there, they're stable, but epigenetics is going to allow modification of how they're read out and interpreted by the body, and therefore how you might generate certain types of cells or generate certain types of behaviors down the line. So my lab right now at Bowdoin, we do a lot of work trying to understand what is it about early life adversity that changes the way our genes are read, and specifically how a gene that codes for an inhibitory cell, meaning a cell that tells other cells to quiet down, how that cell might be impacted. And that cell that we look at is parvalbumin. The name's not important, but the function is, especially in areas of the brain that help us to inhibit behavior, right? This is going to allow for behavioral inhibition. And so we can look at this through DNA methylation, which is where we actually are adding a methyl group to DNA, which makes it harder to read. We have these things in our body that allow for reading of this blueprint that is our DNA, and we can block that with experiences adding these methyl groups to our DNA. So it's not going to read it the same way that it typically would. And if we look at, so here are just some examples, because I can't do any presentation without a pretty brain picture. We have parvalbumin, which is going to be on your left, those nice pink cells, and then we have on the right 5-methylcytosine, which is a marker for DNA methylation. And so this would be localized in a cell nucleus where the DNA is. And we can look at an overlay of this specific cell type that I'm interested in and changes in methylation and actually quantify to a certain extent how methylated is that specific cell. And from that, we can try and understand, is this cell individually impacted? Are there other cells that are equally impacted? And we can correlate this with behavior of our animals to try and understand whether this helps to drive that behavior and whether we can mitigate it. And most importantly, when we're thinking about our environment is changing the way that our genes are read, another thing to remember is that our environment is also constantly changing. Our genes, the methylation on them that's reading them out is incredibly dynamic. So there's a huge opportunity here when we're seeing this very early on to mitigate things, especially during childhood. So there's some things here that are going to allow us to explore some of these the substance use more critically at the genetic level, and others that I think bring us a lot of hope that we can kind of mitigate things much earlier when children's brains are still really receptive to changes and support. So that is what I have for you, and thank you.
Well, good morning, everyone, almost afternoon. My name is Vivek Kumar. I'm also a faculty at the Jackson Laboratory, and I am a geneticist, like most of my colleagues. And as my slides come up, I'll just say I grew up in Tennessee. I did my training in Illinois, California, and I lived in Texas, and I've been in Maine for the past 8 years. And I think the leadership in Maine in dealing with the opioid crisis has been exceptional. I really echo the keynote speaker. And this is a direct result of Governor Mills and led by Director Smith. So I really want to thank them for organizing this and to show the leadership. So, we talk about genetics of addiction. I'm here to give you a clear example of how our genes are going to affect behavior. So I'm at the Jackson Laboratory, which is, as you know, beautiful Acadia. And let me start by just talking about the addiction cycle. And I was actually looking in Journey magazine, which is in your blue bags, and there's a nice article on the addiction cycle. So addiction starts with social intake of drug. We go out, we have a drink every Friday night. Soon that kind of escalates into compulsive drinking. We drink too much on Friday night. Sometimes we start drinking on Saturdays and Sundays. And pretty soon what happens is we reach dependence. Our brain essentially gets used to the dopamine release every time we're drinking or consuming the drug of abuse. We realize this, and the person may then want to quit, and they may stop taking the drug. That leads to withdrawal because the brain is used to getting that chemical, and now it's not functioning very well because it doesn't get that chemical. So that's withdrawal, and then that will essentially lead to a relapse. So the person then goes back and starts consuming the drug again. Now, some individuals will have protracted withdrawal where they are able to stop for a long period of time, and some will go into recovery. So this is essentially the cycle of addiction. And every part of that cycle is controlled by our genetics, our environment, many things like stress, like adverse childhood events, and the things that you heard, conditioning, all these things kind of play into this. And what we can do is all of those conditions are really hard to study in the human, but we can study them in model organisms, such as a mouse. A mouse and humans share most of our genetics. We have very common brain structures. Obviously, a mouse's brain is much smaller, but all the essential circuits, all the essential cell types, they're all there. So we can then control the mouse's environment, its genetics, to really study all those different phases of addiction. And yeah, so a mouse is a really model organism-wise, allows us to manipulate the environment, manipulate its genes, and we can kind of break down a really complex disease, which is a substance use disorder. Okay, so one of these mice are on cocaine. Do I need to tell you which one? So here's, we just injected a mouse with cocaine. We're looking at kind of the initial response, the first response of the animal to the drug. And when we do this experiment, we find that the mice become hyperactive. And so we did this experiment on a set of mice, and these are, I'm going to just call them Maine mice and Maryland mice. So in 1951, so these mice are essentially inbred, meaning they're clones of one another. So we have hundreds and thousands of inbred mice in Jax and in Bar Harbor, and we shipped in 1951 some of these mice from a particular strain to Maryland, and they were exactly identical. Imagine, you shipped a bunch of your pet mice, they were exactly clones of one another, to your friend in Maryland. The friend in Maryland starts to maintain them by breeding them, and then we maintain them in Bar Harbor by breeding them. And breeding, as we know, there's some errors that are kind of introduced. This is known as genetic drift. This is how evolution kind of creates fodder for selection. So in 1951, these mice are exactly identical. They're just moved from one location and kind of, let's say, split. And so in 1951 is Harry Truman, and American in Paris is kind of the big winner in the Oscars. And in 2013, we looked at these same mice in Maine and Maryland. We just got them into a common location. And we looked at that initial behavior response, that running around after you inject them with cocaine. Now, they were exactly identical in 1951, and we're looking at them many years later. And it turns out that the Maryland mice actually respond less to the drug, cocaine. They run around less, okay? So everything is identical. They're picked up some mutations, changes due to genetics, and now we can do what we like to do, which is, as a geneticist, we can map this difference. So I'm going to save you about 2 years worth of work. And we did genetic mapping, just like Dr. Chesler showed through that kind of just setting up those crosses. And it turned out that the Maine mice have this sequence here, GG, CTC, TCC, CCA. And the Maryland mice have one nucleotide change. That TCT in the middle is changed to a TTT. And that changes one amino acid in a very particular protein, from a serine to phenylalanine. And it turns out that that one change leads to that difference in cocaine response. Okay. And this change was picked up just by pure chance in the mice that were bred in Maryland. Okay. So that's all great. So this now gives us a real molecular handle. Now we can go and we can investigate a pathway, we can investigate a protein, and we can really have mouse models that can be shipped around to different labs to study. And that's really the power of having kind of this, this resource. So we did that. So we made mice that, had this mutation. So we essentially created new mouse models. And then we shipped them to our colleagues that study alcohol addiction, our colleagues that study nicotine addiction, and our colleagues that study food binging. Now, we all know, we've heard this polysubstance use disorder. There's a whole table on co-occurring disorders on the right over there. So, we wanted to look to see, well, does this mutation affect other drugs of abuse or even natural rewards like food? Before I get to that. So finding the one mutation. So finding the one mutation is like finding, one person out of the combined population of India and China combined. So this is the mouse genome is roughly 2.8 billion base pairs, and we found that one. So this takes a lot of technology, genomics, genetics, which are all kind of resources that the community has. Okay, so then we took the gene, we shipped our mouse models to different locations, and our colleague in Virginia looked at nicotine, and it turned out that the same gene regulates nicotine reward. Our colleague in University of Maine here in Orono looked at alcohol, and it turns out that it affects alcohol drinking. And a colleague in Boston University looked at food binging, and it turns out that the same gene affects food binging. So why is that? It's because the underlying circuit, the reward circuit, is common. That reward circuit did not evolve so the mouse could take cocaine. It really evolved for natural reward. Natural rewards are food, sex, social interaction. And we know this in recovery, right? So if someone is addicted to a drug, you want to substitute that negative behavior with something positive. That you feed that circuit with exercise. You feed that circuit with natural kind of socialization, positive socialization. Yeah, so that's pretty much the message I have. So we're essentially have these kind of really good tools to study mechanism. We can then start to target specific pathways for treatment. But it also tells us a lot about how the neural circuits work and how the neural circuits lead to multiple this polysubstance use disorder, and also natural overeating or not socializing. Yeah, that's all. Thanks.
I'd like to thank the organizers for having us here today. This is my 5th year attending the Opioid Summit, and I'm always intrigued and encouraged by the amount of people who care about the issues that we care about coming together in this one venue. My name is Jason Boubier. I'm a research scientist at the Jackson Laboratory. I got my undergraduate degree from the University of Maine at Farmington, so go UMF Beavers. And during that, after I finished my degree actually there, I was able to spend the summer following that year at the Jackson Laboratory. So I actually was selected by the Mills family in Farmington to have an internship award in which I was able to spend the summer working with brilliant scientists at the Jackson Laboratory. And after working there for one summer, I knew I was going to come back. I went to graduate school, got my PhD, and came back and have continued to find a way to help advance different diseases, currently focused on addiction. And I've been working with Dr. Chesler at the Center for Addiction Biology. And one of the things that sets my program apart from theirs is that I'm not just looking at addiction, but I'm actually looking specifically at overdose. So overdose as a physiological effect on the organism. So when somebody dies or administers an opioid, they're going to suffer a variety of things, including opioid-induced respiratory depression, so how their respiratory rate goes down. But there's other things that occur when you have an extreme dose of an opioid, like your larynx starts to close up. Another thing that can happen, especially with fentanyl, the synthetic opioids, is a wooden chest syndrome develops, in which there's muscle rigidity in the diaphragm. And these things are able to be reversed pretty well with Narcan, especially for the endogenous or native opiates like morphine or heroin. But these synthetic opioids like fentanyl, carfentanil, and now the combinations of things like fentanyl plus xylazine for the tranq, that's not really going to be helped by Narcan. We need something else to help us in these situations. And then there's a new drug I heard about coming out of Cleveland, protonitazine, I guess, and it's really independent of the opioid receptor totally, so it's not going to involve or be solved by Narcan at all. So we need to find alternatives to alternative reversal agents, and different ways to do that is to use this mouse model system that we've been talking about. So why are we using mice? Well, the similarity of mice to humans, about 92% at the DNA level, Fruit flies, it's about 44%, yeast 26, plants 18, and chimps or primates up to 98. And then the human populations amongst ourselves, we're very similar to each other, but we have some diversity. We're 99.9% similarity. And so there can be these single nucleotide polymorphisms like Vivek was showing with his Maryland and Maine mice that can be floating around in the population that give us our different compositions. So using this same scenario or this population of mice, at the Jackson Lab, I have a group, they're all musculus, they're all mice, but they're different strains or different inbred lines. And so that's represented on the left here with the different colored mice. And they all respond differently to drugs. And one of the things that I found in studying morphine, for example, for overdose, is that amongst 8 different strains of mice, there's about a 4-fold difference in their tolerance to morphine, a natural opiate. But amongst those same 8 strains of mice, there's a 150-fold difference in their sensitivity to fentanyl. So what that means is that if you go to use a dose for the first time and you have no idea what your susceptibility is, you don't know which of these populations on the right you fall in. Is your respiratory depression going to be so sensitive to the opioid that one dose is going to be fine, that you could take a huge high amount, like up in the top right? Or are you very sensitive where you one small dose that's a fraction of what your friend is using is too much for you, and you're very sensitive. So what we have here is a natural genetic variation in the population of mice that can help us understand the natural variation that's occurring in humans. And by looking in the population, we can identify what has Mother Nature set up for us as possible targets to develop new therapies. What is there that's different amongst these people or these mice that we could possibly develop a new drug, something that's going to help us to stimulate respiration, but let the people still maintain the euphoria or analgesia of the opioid, but not have the respiratory depressions held down. So, be able to maintain their high but not die from the respiratory depression, have their respirations uncoupled from the analgesia. So we can use this mouse population kind of to help us find these genes in humans and target those for development. And I'll turn it over to Mike.
All right, excellent. Thank you very much, panelists. Fantastic introductions. We have time for some questions, and some people have already been very responsive to the Slido opportunity. We'll just leave that up there, so if anything pops up, please submit. I think where I'd like to start is with education. Jason, you brought it up in your remarks. That's kind of how you became interested in doing science based on the fellowship you got at the University of Maine Farmington. So I'd love to hear from Jennifer your perspective of how you're engaging students in research from the point of view of the college, but then I'd open it up to one of my JAX colleagues for how you have engaged students in your own research. So how is education a way into creating scientists to help, and other professionals, right, to help address this issue?
It's on?
Yes.
No? Yes?
Yep.
Okay.
Speak right into it. It's on.
Perfect.
Yep.
So I actually, just to answer that question, I think my own experience getting into science is probably relevant here. So I went to Colby College, which is also up the street, while I was there, I worked in a lab that was funded by Maine INBRE, or the Idea Network for Biomedical Research Excellence, which gave me a lot of opportunities to work with a new investigator and to really realize that research was something that I loved and that I could answer questions at a really basic level that had translational ability far further than we could even really see in the lab. And this is coming from somebody who's a first-gen student who had no idea really what I wanted to do or that neuroscience was even an option for me. Now at Bowdoin, I'm actually funded by the same grant that I benefited from as an undergraduate, and so we're funded through Maine INBRE, which is a program through the National Institute for General Medical Sciences through the NIH. And that provides us funding to answer some questions that are pricey because research is extremely costly, especially the kind of research that involves looking genetically or epigenetically to answer some of the questions that we have. And so at an education level, I think the availability of resources has been huge in allowing my students to investigate questions that are going to matter to both them and to people that they care about, and getting them hooked on something that they feel is relevant in their own lives has been huge in getting them excited about continuing in research.
Great, thanks. Vivek, would you care to follow up on how you integrate students in your lab?
Yeah, absolutely. So my journey in science also was because I was given opportunities to be in a research lab. When I was in high school, I started with science fair projects and going to University of Memphis, where I grew up, and doing research there. And that really changed my whole perspective. I mean, just like drugs of abuse at a really young age can be bad, science at really young age can be really good. So I disappointed my parents by not being a medical doctor and pursuing a PhD. But I think, like that early experience made all the difference. And, I'll also add one more thing, which is almost all of us here are funded by the National Institute of Health. So we're taking money from the Health and Human Services Department to do research. Part of our mission is to go out and educate the next generation of scientists. That is just one of the most important things. If we can't talk about our research and to the lay public and kind of educate the next generation of scientists, I think we're doing a disservice by taking all this money just to do really cutting edge but not understandable research.
Thank you very much. So Alyssa, I'm going to direct the next question to you. And the question is, and this is, I think, probably in many people's minds, are there reliable genetic tests for predicting addiction in humans, and how accessible are the tests to the general public?
I think the answer to that is not yet, and possibly, unfortunately, maybe not ever. Addiction, we've shown you some pinpoint genes that influence behaviors, and we find them when we control the genetic backgrounds. A lot of times when we look at very simplified genetic backgrounds, we have so much variation in our life experiences, and we have so much variation in our DNA. And if you think about something as complicated as the brain, it's influenced by thousands of genetic variants. So, and for those of you who've been on some genetic websites, you can see elevated risk from everything that we know about the genetic variants that influence some of these traits. But it's not explaining so much of the variation that we can say for a particular individual that this is what's going to happen, or how high their risk is. We may one day be able to do that. It's a known issue that most of the existing genetic studies have been done primarily in people of European descent. There's a big push to diversify the populations that are being studied in human genetics. So I think that the challenge is we don't really know, but we do know from our genetics work that there is biology there. We're identifying individual genes that influence that biology, and we're finding ways to use the information from those genes to think about therapeutics and to think about how the circuitry of addiction vulnerability varies in different people. And so we can learn quite a bit from genetics, but we can't yet test people and say, you're safe, you're not safe.
Just a follow-up, Alyssa, if you, there's lots of consumer-based genetic tests available in the market, 23andMe and many, many others. So if you are presented as a consumer with a test that can answer that question, what would your response be?
The question, I guess you're asking, how would I use that information?
Well, no, I mean, I guess what I'm saying is, should the buyer beware?
Yeah, I think those sites are very entertaining. I'm interested in whether or not I can taste cilantro. I know how I respond to cilantro, but I can see that 23andMe also knows. They're often useful, and I think about it as I could use that information to prioritize. and say, okay, I'm not that great. I'm not going to eat healthy and exercise and take all these precautions and get all my regular medical screenings, but where should I prioritize my energy? I might use that information for that. These are emerging technologies, and I think they're maturing quite a bit. But again, those are statistics applied to a total population, and a population that's composed of people who sought out 23andMe and got their DNA tested, and there's other companies as well. I don't want to pick on one. So the question is, how do we go from that population average to what's happening in one person? And that's a very challenging thing. Those averages don't necessarily explain what's going on in one person based on all their other genetic variants, based on their entire life history. So again, it explains such a small fraction of your risk. It might say I'm 2% more at risk than one of these people are, but it doesn't. I can't use that 2% to do anything important. I hate to be so negative, but I want to say, we have to dial back our expectations of individualized prediction for something as complex as these behavioral phenomena that involve so much environmental influence and so much exposure history influence.
Thank you. Let's stick with individual genetics. And Jason, I'm going to ask you to take this one. If you can identify genetic predisposition to addiction, can you also identify genetic predisposition to likelihood for recovery? Are some people more genetically set up for success in recovery?
Wow, that's a great question. I have not actually studied that myself, and my guess is that the trait would be equally as penetrant in recovery as it is in development of a use disorder. I don't know if anybody else here has talked about it.
We've been starting. So the first question is, how do I study in laboratory mice something that looks like recovery? And we have some traits that we're looking at. And then the question is, how do I simulate? We talked about, other behaviors that we might want to engage in instead of substance use. The problem in addiction is that compulsion to use the drug. That's the behavior that is being elicited in response to all of life. And so the question is, how do you break that pattern? What other patterns could be reintroduced, or what other patterns can an individual form to reshape the balance of their life? And how do I study that in the lab? And so we've been working on ways of doing that type of study. And then the question is now, if I get that to work, how much is the response to that explained by genetic variation? Now, we do look at, if I take the drug away, does the mouse still seek the drug, or do they move on? And I can tell you that trait, extinction, varies quite dramatically. And I can also look at something that's almost like relapsing, which is now if they have access to the apparatus again, do they press the lever, do they reinstate that seeking behavior? And again, that's genetically variable. So yes, we think there is genetic variability in propensity to change behavior following acquisition of this compulsive drug use pattern. But we're in early stages of identifying what genes and what the biological basis of that change in behavior is.
So I think of the whole process from a neurobiological perspective as one of plasticity. So absolutely, plasticity is important for breaking the system. It's also important for restoring the system. I gave a talk at a recovery event in Ellsworth, and someone used the analogy. They said, if you think of the brain as like a piece of paper and addiction as crumpling up that piece of paper and recovery as kind of smoothing it back out, you never fully can get rid of all the creases, but yeah, you can, fairly go back to normal with the right treatment and with the right kind of holistic approach. Certain insults are much harder to turn back the time on than others, like what Dr. Honeycutt talked about in terms of early life changes, because those kind of fundamentally change circuits, connectivity maybe, and those may be much harder to recover from, but later life changes may be easier to recover from. Someone who's been using the drug for 2-3 years, it may be easier to recover than someone who's been using the drug for 20-30 years.
That's a great transition, because a member of the audience asked the question for you, Jennifer, to what extent is it possible to reverse the early childhood experience? What have you observed in your lab?
Yeah, so that's a great question, and it actually kind of goes along, I think, with some of what was just said. We do focus a lot on risk, of course, because we're trying to answer a question and really trying to inform treatments. But we also have some research on resilience. We can see in the data that not everyone who utilizes an illicit drug will go on to develop a substance use disorder. There are, much like that, if you look at populations of children who have grown to adults, not everybody who's experienced a significant number of adverse childhood experiences is going to have the same outcome. A lot of that has to do with early intervention. So that is a huge piece of that puzzle. When we're thinking of early intervention with children, this often looks like kind of mitigating at the behavioral level, going in, providing resources for kids that maybe are not in a stable environment, and streamlining some of those resources. When we're looking at early life adversity, and from my own research, we see that the brain can actually develop differently while an individual is experiencing. In this case, of course, we're talking about rats, but we see this in humans as well, that were once institutionalized in terms of brain connectivity. But if you intervene early on at various levels, it could be at the behavioral level in humans, it can be at a biological level, for example, in model systems, we can see some kind of mitigation of that, but the biggest piece, and as I mentioned when I was speaking, was that our brain is very plastic, and the changes to the way our DNA is read is very dynamic, and is constantly kind of changing. So it's not just at one point that we want to have this. Of course, when your brain is extremely plastic and these things are occurring, is kind of the best time to address these issues. But of course, later on in terms of treatment, this is also a really important piece of that as well.
Thank you. So I'm curating questions up here. There are way too many to get through. They're all great. But it's a reminder that if you are really interested in having your voice heard, come right down front and we'll take a question from the floor. Don't be shy. Thank you. Okay. So Jennifer, staying with you, somebody was really paying attention to your presentation, and they asked the question, were there other stimuli in the experiment other than the cocaine? Describe the environment a little bit.
Yeah, so in that particular experiment, it was a conditioned place preference. You pair a single experience with cocaine with a specific room. And then you pair another room with a different type of drug, and or in our case, we just gave them salt water, right? Just saline. And then you ask them later on, which room do you prefer? So our animals that went into that had experienced early life adversity kind of overwhelmingly wanted to choose the cocaine room. A caveat of that, and what was mentioned as well earlier, is that we also want to understand this in the context of sex. And so that particular piece of data, we did that experiment in both males and females. We saw that effect in our males, but we did not see that effect in our females. And what we also saw is that could be mitigated by kind of blocking certain pathways that we think might be involved in kind of that more risk-taking behavior following early adversity that tends to be a little bit more male-specific in terms of risk-taking versus more kind of anxiety-specific in some of our female animals.
Thank you. Okay. Oh, we do have a question. Fantastic. Go ahead.
Hi, good afternoon. I'm wondering if there are any current is there any current research regarding expanding neuroplasticity? A number of you panelists have been talking about the importance of neuroplasticity with regard to addiction and resolving addictive behaviors.
Yeah, so it's one of the most active areas of research for the past probably 20 plus years in neuroscience. If you go home and you remember anything about what happened here today, it's because your brain has literally physically changed. So I think one of the more exciting areas in neuroscience right now is psychobilins and mushrooms and how they lead to kind of extended changes in plasticity in the brain and how people are using that to treat PTSD and other kind of even substance use disorders. So there's a lot of like exciting stuff on plasticity happening.
Thanks. We'll wrap with one more question. This is the last one. So the question is, well, it's a statement. That's a question. How does this incredible information get reported to the general population in an understandable and effective way? And this is of course part of this, but we're not the general population beyond the borders of this room. So, who does anyone want to share on how public outreach, how engagement is important to the community at large, specifically to substance use disorder?
I feel like I'm put on the spot here. So I write papers. I'm on social media. I've written op-eds for Bangor Daily News, Portland Press Herald, and I'm out here. So
Yeah. Who else? Anything else?
Yeah, I also utilize my family and network of friends, and if they start to ask me about science, I'll go on and to share with them more details than they want to know. But usually they have some really important questions, and I think I may have convinced my 80-something-year-old father that addiction is a disease and not a willpower thing, and so that's a huge step forward for me. So it's small steps like this, having these conversations.
Similar. Social media is a great one. Also, community outreach, getting into your community. I've done a lot of work with some organizations in the past, like Skype a Scientist, where you actually Skype into an underfunded classroom and talk to them about science, and it can range. I think I've had a group of kindergartners, which was probably my favorite, to high school students, to continuing adult education, and it's just a really nice way to kind of really boil down your science to, what are the bare bones, what are people excited about, what's going to get people to just understand a little bit more about how their brain is working.
So yes, we write, we speak, we do programs at our libraries. A few of us work with a local recovery center, Acadia Family Center. We've made Lego models of our brains and of populations and of behavioral equipment and taken them into high school classrooms and tried to teach kids about what it means to have your behavior change, just as we saw with cows drinking milk today. We let high school students press levers and see how their behavior changed when those levers no longer have any action. So we do lots of things to try to put this information in the hands of the public. But it takes all of us going out and getting that word out there in an interpretable and usable way.
Fantastic. Well, I learned a lot today. I am inspired by the hard work of these individuals as teachers, as researchers, as dedicated professionals, leveraging yet another tool in this challenge. So thank you very much for joining us. Thank you for coming.